Good evening, everyone, and welcome to Fortis Memorial Research Institute, Gurugram, for a live session on whole exome sequencing. I am Dr. Shrinidhi Nathany. I am consultant molecular hematologist and oncologist at Fortis Gurugram, and it takes me great pleasure to talk on this intriguing topic of whole exome sequencing. And the word whole exome sounds as if it is a huge thing to do, and although in India we still have a lot of miles to go before we sleep. And so today we are in conversation with two great experts, and I will be interacting with them through a lot of different types of scenarios and questions, which will probably elucidate a lot on what is exome, when should you do exome, what is the Indian scenario of exome, and whether we are there yet, and how far are we still remaining to actually cover that distance. And today I have two experts. One is Dr. Vinod Scaria, and he needs no introduction. And he is one of the most sought after genomic scientists in the country. And he was the first one to sequence the human genome in India. And he is a very renowned personality who knows probably everything under the sun when it comes to molecular biology. And I have followed him since I was a student in molecular biology, and it's like a fan moment for me right now to be being a moderator and asking him questions, and he's on the other side of the table answering mine. And the other one is Dr. Rahul Bhargava. He's the principal director of hematology and transfusion and transplant, bone marrow transplant at Fortis Gurugram. And he also needs no introduction. He is probably the most busiest transplant physician in the country, and he has immense experience. And his newfound interest is in genomics and a lot of multiomics and all different types of hematologic diseases, as well as in fact any disease under the sun. And let's see what they have to talk about exome sequencing today. So next slide. Okay. So my reaction when I saw my guests was a fan moment, basically. So this is just in humor. So now let's get down to some serious discussions, that is exome. So I'll ask Dr. Scaria that what is meant by exome and what is genome, because this is for the knowledge for even lay public who might be watching this, and some general physicians and oncologists who don't really understand what is exome and what is genome. You and I, for us, it is almost normal mother tongue language, but then for others, please can you elucidate?
Yeah, so broadly, of the around 30 odd trillion cells in the human body, we all have a blueprint, and that is what we call as the genome of an individual. The genome that we have is inherited from our parents. Each of our parents contribute to one chromosome each. So you have two pairs, or a pair of chromosomes for each locus. And that is essentially what our genome is. Now, you can sequence this entire genome, which is approximately around 3.2 odd billion bases, which is called a whole genome sequencing. Now, like a whole body MRI, that means that you can read the entire chromosomes, that is whole genome sequencing. But in most applications, you might not need to read the entire genome. You might need to read only small parts of the genome, which encode for proteins, which is approximately around 2% of the genome. And this is what we call as a whole exome sequencing. So a whole exome sequencing reads approximately around 5 million odd bases, or 5 to 8 million odd bases. It's much smaller than a 3 billion bases that you would have in a whole genome. And therefore,
So
it
friends,
looks at only a very small number of, a small portion of the genome, only around 2% of the genome.
So, this is just for everybody, just to reiterate. So, friends, you can understand we have such a huge genome, which is 3.2 billion bases. And if in an anecdote somewhere I read that your DNA is long enough to go to moon 9 times and come back. So, you can understand if you're going to sequence the entire genome, it is going to take a lot of data, a lot of data footprint on the cloud as well. And where would we store all that data? So you do, and you triage that where do you want to do the entire genome, and where you want to do only 2% of the genome, which is exome. Am I right, Dr. Scaria?
Absolutely.
Okay. So moving on, I will just tell you that this is the central dogma of molecular biology. And what he meant by genome was that we sequence the exons, which are in blue boxes, and the introns, which are in white boxes. And that is the DNA. Okay. And if you remove the intervening introns, which are there, which is read as IVS1 and IVS2, if you remove those, then what you get is something called as an RNA, and that has only the protein coding region of the genome. So that is what is called as the exome. So what when you do an exome, you're doing only the exons, and when you are doing the genome, you're doing both DNA and both the exons as well as the introns. So my next question is, some people, they think NGS is different, WES is different. I have a lot of people who come and ask me that, so Srinidhi, are you going to do NGS or are you going to do WES? And I go mad explaining that NGS is the technology and WES is nothing but a broad-based NGS. So I would want you to explain in a very lucid language that NGS is nothing but the technology. So probably I have failed in explaining to a lot of people, but it would be great if you can explain the same.
Yeah. So, if there are clinicians out there, the easiest explanation that you can make is to say that there is something called a CT scanner or an MRI scanner. The CT scanner or MRI scanner is the machine and the technology that enables the scanning to happen. And in the similar way, for the NGS or the next generation sequencing is a technology which will allow sequencing to happen. There are many technologies. The previous one was called Sanger or capillary sequencing. There's a new technology which can scan the entire genome. Now, WES or whole exome sequencing or whole genome sequencing is like a whole body CT scan or whole body MRI scan or just an abdominal MRI scan. So it is just a part or an assay that you want to look for that clinicians use. Technology is
So
much broader. The technology contains much more things that it can do. WES is one application of next generation sequencing.
sure. So, friends, what I want to say is NGS is the platform or the technology or the principle, but WES, or if you're using a 10 gene panel or a 50 gene panel or a 500 gene panel, those are all the tests